Okay, we are going to continue with, I think I really need glasses if I see that here, with Flavia Fontana, the Italian lady from Finland. She works at the drug research program at the Faculty of Pharmacy at the University of Helsinki.
Okay, thank you. All right, good afternoon, everybody. And after some targeting and some liposome, now I would like to present something that is completely different. Today I'm going to talk about the development of Biohybrid Multistage Nanovaccine so far for the treatment of melanoma. First of all, how do you build a multistage nanovaccine? We started in our lab, we have a strong experience on the use of porous silicon nanoparticles, and some of these particles, thanks to their surface chemistry, display immunostimulatory properties. We coated this particle with the polymer. We embedded them into a polymeric matrix by glass capillary microfluidics. Glass capillary microfluidics is a technique that allows a high yield of production of the nanoparticles, up to 700 grams, 1 kilogram per day, which is like the kind of scale that you need for the preliminary clinical trials. It's a versatile technique that allows the production of different particles, and it's easily scaled up thanks to the production of particles by multiple chips in parallel. So we have the final system that is constituted by a polymeric matrix of acetylated dextran coating porous silicon. This multistage nanoplatform then is decorated with vesicles obtained by membrane presenting antigens, tumor-associated antigen, neoantigen by membrane extrusion, obtaining the final system. And here is a SEM, high-resolution SEM picture of the final system. Firstly, we evaluated in vitro how this system would have behaved. So we have here the immunostimulatory effect of the system over peripheral blood monocytes, human peripheral blood monocytes, and we can see that it strongly activates CD86, and it leads to an increase in the production of interferon gamma. So we can see that there is an activation of antigen-presenting cell with an increased expression of the co-stimulatory signal and an increased secretion of the pro-inflammatory cytokines. As was reported this morning by Professor Gallon, one of the new ideas in the treatment of cancer is the combination of two different immunotherapies to prime and boost the immunotherapy against cancer. So first, a cancer vaccine will be administered to the patient to prime the immune response with the production of cytotoxic T lymphocytes, followed by the administration of checkpoint inhibitor. In this case, I'm presenting CTLA-4, that is a molecule present on the T cell, and when is binding to B7 on the dendritic cell, this is like inhibiting the proliferation and the action of the T cell, diminishing the immune response. So with the administration of anti-CTLA-4 antibody, we can allow the T cell to remain active and to have an effect against the cancer cell. The aims of the study were first, as a proof of concept, to investigate the efficacy of this system in vivo in melanoma models, followed by the elucidation of the therapeutic potential of this therapy in vivo. So the model chosen were first an aggressive melanoma model, so B16F10, followed by an immunological one, B16-OVA, to allow for a deeper analysis of the immune contexture of the tumor microenvironment. These were all subcutaneous xenografts. The vaccine was administered subcutaneously in the peritumoral region, and the checkpoint inhibitor was administered concomitantly with the vaccine intraperitoneally. First of all, we evaluated the efficacy in the aggressive tumor model. So we have that 4 animals out of 7 displayed control over the tumor growth after 16 days. with an increase in the percentage of CD8 cytotoxic T cell in the tumor microenvironment. And of this T cell, we had like a huge increase in the percentage of experienced T cell, so the cells that are active and are ready to kill the cancer cell. Moreover, we evaluated whether we could find correlation between the responders among the different groups and the different parameters within the tumor microenvironment. As you can see, in all these cases, so number, percentage of T cell, CD8 T cell in the tumor microenvironment, the state of this T cell, whether they were experienced or not, the percentage of dendritic cell, and how this dendritic cell, if they were activated or not, the responder had higher percentage in all of this. We then moved to check what was the reason why the vaccine was working. And we first evaluated the effect of the different constituents, so the multistage platform and the cell membrane vesicles. And we can see that there is a synergistic combination of the two that controls the tumor growth, in this case, since it is a less aggressive tumor in a higher number of animals. This resulted in an increased number of dendritic cells in all the groups, but surprisingly, in the group with both in the final formulation, we had a lower percentage of dendritic cell compared to the single system. However, this was mirrored by an increased activation of this dendritic cell, as you can see in the last one, that were almost all CD8 and double positive, while in all the other controls we still had immature dendritic cell. We couldn't find a statistically significant increase in the ratio of CD8 to CD4 in this case. And then we moved to evaluate the efficacy of this combo treatment. As you can see here, this is like the standard of care in most cases in melanoma. So the administration of a checkpoint inhibitor, first line. With the priming with the nanovaccine, followed by the administration of the checkpoint inhibitor, we can improve the efficacy of the checkpoint inhibitor and decrease also the area under the curve. I'm reporting the single tumor curves because you can see that in 2 of the animals we had complete remission, while in the other, after a decrease, we had again an increase of the tumor mass. So probably another boost of the vaccine or another injection of the checkpoint inhibitor would have been needed. We then evaluated how was the tumor microenvironment. So we could see an increase in the percentage of the dendritic cell. Again, activation of the dendritic cell with increased percentage of the double positive dendritic cell. And in this case, a huge increase in the percentage of CD8 cytotoxic T cell within the tumor volume. So in conclusion, we could achieve efficacy in an aggressive model with a control over the tumor growth in approximately half of the animals. There were significant changes in the tumor microenvironment with an increase in dendritic cell and CD8 plus T cell. This vaccine was efficacious also in immunogenic model, B16-OVA, with a proven synergistic effect of the components of the system and an increased maturation of the dendritic cell. We could prove an enhanced efficacy in the combo treatment by vaccine followed by immune checkpoint inhibitor with control over the tumor growth and, in some cases, remissions. And this synergistic effect with the immune checkpoint inhibitors was due by an increased presence and activation of dendritic cell and increased percentage of CD8 T cell. Finally, I would like to acknowledge my supervisor, Professor Santos, our collaborators, my lab, and you for your attention. Thank you.
Thank you very much. From the audience, any questions? Okay? Let me see. Maybe you can manage this way.
Thank you very much for the nice talk.
Thank
I
you.
was wondering, is there also synergy observed if you use chemotherapy along with the vaccine, which might be happening
yes.
also quite often?
We haven't tried yet. It's still in the initial stages. We have different combinations that we are planning to try, also with other checkpoint inhibitors and having maybe chemotherapy, vaccine, and immune checkpoint, the triple combination. Thank you.